Now, what happened is the messenger RNA. This is now the process messenger RNA is translated into protein. Okay? It is the process where ribosomes synthesize proteins using the mRNA transcript. It involves changing the information from the nucleic acids to polypeptides. As you can see, the messenger RNA strand or transcript, it is made up of nucleic acids or nucleotide. Nucleotide. And this nucleotide, they are the one that is translated into protein, to polypeptides. When you are talking of polypeptides, this is a chain of amino acids who are the key players in translation. The messenger RNA, the transfer RNA, the ribosomes, and the amino acids. The coding regions of the messenger RNA, you call that codons. And these codons are the one that is translated into one protein by ribosomal RNA. And it begins with a start codon, AUG. Do you know what is the amino acid that is synthesized by this codon AUG? Anyone?
Methionine po.
Okay, methionine. When you are talking, look at the codon. The codon is made up of 3 nucleotides: adenine, uracil, and guanine. The codon. And do you know what is the anticodon of AUG? The opposite, the opposite of codon is the anticodon. And you know what is the anticodon of AUG?
UAC po, Doc.
UAC.
UAC.
What is that UAC? UAC.
Tyrosine. Tyrosine po, Doc.
What is that UAC?
Tyrosine. Amino acid.
That is tyrosine, but that is uracil, adenine, and cytosine. That is the anticodon. But it is coding for amino acid tyrosine. And again, there are also stop codons, which is responsible for ending the translation process. And what are these stop codon? They are UAA, UAG, UGA. In the case of this messenger RNA strand, you could see the stop codon, which is UGA. And the start codon is the starting point, and that is the AUG. And the stop codon, this is responsible for termination or stopping the protein synthesis. When, when translation reaches the stop codon, protein synthesis will stop. And then each codon encodes for a specific amino acid. Each codon encodes for a specific amino acid except the stop codon. What is the steps in the translation process? Initiation. Okay. First, there is a step or initiation process wherein the mRNA enters the cytoplasm and become associated with the ribosomes, which is the start of the information it encodes. And then there is the initiator transfer RNase. The transfer RNase is the one that initiate the translation process containing the anticodons, which pair up with the messenger RNA codons inside the ribosomes. And so the start codon is AUG, which is coding for methionine. Okay, this is the small ribosomal RNA, and this is the one, this is the transfer RNA. Take note that the transfer RNA is the one that is carrying the amino acid, and also below is the anticodon, wherein the codon of anticodon UAC is AUG. And this is the large subunit of the ribosome. And this is the small subunit of the ribosome. The large subunit of the ribosome, it has binding sites. And you call this the A site or the amino acid group or the amino acid binding site. It is the transfer RNA is bound into the A site carrying the amino acid. That's why it is called amino acid. And in this case, this binding site is the P site. And this P site is known as the peptide or the peptide amino acid. That means that this is the binding site where the transfer RNA carrying the polypeptide chain will be bound on the P site, the peptidyl site. And the E site is the exit site. When this transfer RNA will transfer to the exit site, that is where this transfer RNA will be released or exit from the large subunit of the ribosome. That's why it is called E site. Okay, so this is now the complete setup of the translation process, which involve three transfer RNA: the ribosomal RNA, the transfer RNA, and the messenger RNA. And this initiation factor, 1, 2, and 3, this is very important in the translation process, because without this initiation factor, IF2, IF1, IF3, translation will not happen. Translation will not happen. So, as you can see now, that the three ribosomes are combined. Okay, here is now the transfer RNA carrying an amino acid arginine, and it has an anticodon known as UCU. Anybody know what will be the codon of this anticodon UCU? What is the codon?
Hello po.
Yes.
Doc Sering.
What is the codon?
UCU po.
That is the anticodon.
Apo. Sering, Doc.
What is the codon of UCU?
Ah. Ah.
Nasa harapan mo na. Always think of complementary bases.
Sir,
Who is talking?
AGA.
Huh?
AGA po.
AGA.
GA.
This is the one. Can you see this one?
Yes, Doc.
Okay, that is the codon of anticodon UCU. That is AGA. Uracil is complementary to adenine, and cytosine is complementary to guanine. Again, uracil is complementary to adenine. The complementary base pairing, always think of that. Okay, so that is. So, now the methionine, what happened to the methionine now? The methionine from the A site is now transferred to the P site. But what is the codon of UAC? What is the codon of UAC? Anybody?
EUG po, Doc.
Huh?
AAV.
What?
AUG.
AUG. Bakit nandito na siya ngayon sa? Bakit nandito na siya sa GGU? What do you think? How come nandito na siya sa GGU? Anybody, why? How come nandiyan na siya sa GGU? Yulimel, are you present, Yulimel? Yulimel, are you present? Jem, is Yulimel present or absent? Kenneth,
Sir, she was present po.
biglang nagkaroon ng lost connection? Kenneth,
Yes, Doc.
yes. Did you get my question?
Hindi po, sorry, Doc, kasi I'm working. I'm working now, so. What's
You
the
are
question
working
again?
where?
In a hospital.
You are working.
Yeah.
Nagsaswab ka ba dyan? You are listening to me? Are you swabbing?
No, I'm not swabbing, Doc. I'm purely in the laboratory. Inside.
Laboratory.
Yeah.
Ano lang, tinitingnan mo lang yan? Are you just looking at?
So, what's the question again, Doc? If I may ask.
How come yung GGO is GGO is the codon of UAC?
GGU is the codon of AUC.
Is that the codon of UAC? GGO. Can you see that?
Yes, Doc.
Is that the codon of UAC?
No, Doc. It's not the codon of AUC. Because
Oh, what is
supposedly
the anticodon? What is
the
the codon of UAC?
UAC,
What is the
AUG.
codon of UAC?
The codon for AUC is AUG, the start codon.
What is that start codon?
AUG or methionine.
AUG. And how come nandiyan siya sa GGU? Anong ibig sabihin yan? Why? How come the transfer RNA is in the GGU now? Anybody who could answer that question? Just a guess. Just just a wild guess. Wild guess.
Sir, I have a wild guess
Yes,
po.
yes. Who is talking?
Gem po.
Jem? Okay, Jem.
Yes, sir. There is a gene mutation.
Okay. What? Jem, are you still there?
Yes, sir.
Oh, anong sagot mo? What is your answer?
There is a gene mutation po.
Wala pa tayo dyan. You just look at the messenger RNA. Anong ibig sabihin yan? What happened to the UUC? What happened to the CUU? What happened to the AGU? What happened to the GGU? All of these have already been translated into protein. It has been translated into protein. Ang ibig sabihin lang ito na lahat yan na-translate na into protein. And so, now, so what will happen to this arginine? It will what? It will now bind into the A site. It will bind into the A site. And then what will happen to the methionine? Okay, what will happen to the methionine? Then, what will happen to the methionine? It will be transferred to the E site. But what happened to the amino acid that this transfer RNA is carrying? The amino acid will be transferred to the transfer RNA that is carrying the amino acid arginine, which will start the polymerization of the amino acid. Nanggaling, he bind into the A site and then transfer to the P site and then transfer to the exit site. And then what will happen to this transfer RNA? And then here, another transfer RNA will bind into the A site carrying the amino acid alanine. Okay? As you can see, class. And then what happened to this transfer RNA that previously is carrying methionine? It is released from the E site. And then what happened to alanine? What happened to this transfer RNA? What happened to this transfer RNA that is carrying the two amino acid? This will again transfer to this one, and then it will exit. And this one will transfer to the P site, and then here comes again another transfer RNA. That is why this was already coded. You see, This one is also coded CUU. And so this is now what happened to this one. Okay, these were already coded this codon. In other words, there were a lot of transfer RNA that already passed through this binding site. So, like for example, methionine. The methionine, the code is AUG. Phenylalanine, the code is UUC. CUU, the code is leucine. AGU, the code is serine. GGU, the code is glycine. AGA, the code is arginine. GCU, the code is alanine. And GUA, the code is valine. In other words, there are already how many transfer RNAs that pass through this ribosomal RNA? There were already 1, 2, 3, 4, 5, 6, 7 transfer RNAs carrying this amino acid that pass through this ribosomal RNA. And so there is now polymerization of the amino acid. And here comes another one. Alanine. Transfer RNA that is carrying alanine. Okay? That transfer RNA binds into the A site and then transfer to the P site. And the polypeptide chain that is being carried by this transfer RNA was transferred to this transfer RNA carrying the amino acid alanine. And so after that, translation will not proceed because the next codon is the stop codon. So translation will stop because a protein will bind into the stop codon, causing the stoppage of the translation process. So it will stop. And these are the stop codons. And then after that, what will happen? All the structures responsible for the translation process will be released. Like the transfer RNA, this is now the polypeptide chain. This now the polypeptide chain will be released. The large ribosomal subunit will be released also. The small ribosomal subunit and also the messenger RNA transcript. It will be released. And here is now the protein. Okay? So that is what will happen if the translation will stop. There is dissociation of all the components from the ribosomes. And this is now the polypeptide. This is now the polypeptide chain, the newly synthesized protein. The polypeptide may be subjected to further processing before assuming the three-dimensional configuration necessary for function. Do you know where this protein will be processed? Do you know the processing of this protein? Where this protein will be processed? Do you know? Anyone? Jem, do you know where this protein will be processed? Gemella?
On the ribosomes po.
On the ribosomes. Doon ba pinaprocess ang protein sa ribosomes? Ever? Joyce? Do you know where this protein will be processed and stored?
In endoplasmic reticulum, Doc.
Is this Joyce?
Yes, Doc.
Yes. Jem, you have to go back to our lecture on the role of the endoplasmic reticulum. It will be processed, it will go to the endoplasmic reticulum, where it will be processed and then transported into the Golgi apparatus, and then from the Golgi apparatus, it will be transported out of the cell, outside of the cell. All the proteins that make up your body in the cells, they are made this way. And we are talking of a molecular level. Like, for example, when you eat meat, when you eat egg, when you eat soya beans, which is rich in protein, this is how it is being processed. Okay, class, do you have any question? Do you have any question, class? Can you see this class? Hello? Hello?
Yes, sir. We can see po.
Can you see this slide?
Yes, Doc.
Okay. Now, we're going to discuss about how the DNA is being repaired. When the DNA, for example, is affected by mutagens. It is affected by ionizing radiation. It is affected by chemicals, strong acid and base, X-rays, UV light. So just imagine yourself. You have been exposed to different types of chemicals. Ionizing radiation. You're exposed to ionizing radiation. You're exposed to X-rays. You are exposed to strong acid and strong bases. And yet, you do not develop cancer. And yet, you do not develop genetic disorder. And yet, your body is normal. Even how much you're exposed to the environment. Like, for example, Jem, do you hear me?
Yes, sir.
Have you seen Jem? Have you seen Jem? Anyone have not yet seen Jem? All of you have seen your classmate Jem?
Yes, Doc.
Gemella? Huh? Angel?
Yes,
Angel?
po. Yes, po,
Have you
Doc.
seen Jem in person?
Ay,
Gemella?
not yet. Only in the picture po, Doc. Picture lang
Only
po.
in the picture. But you should meet Alexander. Alexander,
Yes, Doc. Yes, Doc.
have you seen Jem?
Not personally, Doc. I have only seen the picture.
Okay, you go to PGH if you want to, or the Philippine General Hospital, PGH, if you want to see Jem in person, so that you could appreciate the beauty of Jem, which is flawless. Although Jem is fond of going to Boracay and then exposing herself under the sun. You can just imagine. Huh? But in spite of that, sunbathing, Jem is fond of sunbathing. Can just imagine what will happen to the DNA of Jem. Can just imagine what will happen to the DNA, what will happen to the DNA of the skin of Jem. But in spite of that, the skin of Jem is still flawless and very smooth. Jerel, Kenneth,
Oh, yes, Doc.
have you seen the face of Gem?
No, Doc.
Not yet. Okay. Gem, is it true that you are going to Boracay?
No, sir, I don't travel that much.
Okay, but you have been to Boracay.
Not yet, sir.
Okay. So, in spite of that exposure to sun, the face, the skin of Gem does not change. There is no mutation. Gem did not develop cancer, skin cancer, skin diseases, abnormalities, and the primary reason why because of this DNA repair mechanism. That any abnormalities that will occur in the DNA will be repaired naturally by our repair mechanism, built-in repair mechanism. And the DNA repair, it is a correction process of the spontaneous changes in the DNA, which cause what is known as accidental lesions that happen continuously in the DNA strand. You can just imagine the UV light entering your skin, is bombarding your skin when you are exposed under the sun. It causes breakdown, breakage, fragmentation of your DNA strand. Okay? And this usually, as what I am explaining to you, that accidental lesions, accidental lesions is brought about by heat, metabolic accidents, radiation, like for example, ionizing radiation, alpha rays, gamma rays, and exposure to substances in the environment, like like toxic acid, toxic gases. And for instance, 1 in 1,000 accidental base changes to DNA results in permanent mutation, and the rest are eliminated with remarkable efficiency by DNA repair. This is what happened. What is the significance of DNA repair? It is very important in the DNA repair of enzymes, protein. If the DNA is repaired, the proteins that will be synthesized will be normal. You can just imagine if the DNA has mutated, if there is a change in the DNA structure, you have to go back to transcription and translation of the DNA. When the DNA is transcribed and then it is translated into protein, what will happen to the protein that is synthesized? It will be abnormal. And when it is abnormal, the protein or the enzyme could not be used in the normal metabolic processes. It also shows an increased rate of mutation nor an increased sensitivity to DNA damaging agent that follows the inactivation of DNA repair gene. If there is no DNA repair, there will be defects in human gene that normally functions to repair the mismatch base pairs, resulting to some DNA replication errors, leading to an inherited predisposition to certain cancers, which reflect an increase in mutation rate. Without DNA repair, a human disease called Geoderma Pigmentosum, wherein the afflicted individuals which have an extreme sensitivity to UV radiation because they are unable to repair certain DNA photoproducts, which led to serious skin lesions and an increased susceptibility to certain cancers. This is what happens if there is no DNA repair mechanism in our body. What is the consequence? Again, without DNA repair, consequences. We know that DNA is a complex organic molecules that is susceptible even to normal cell conditions, to spontaneous changes that will lead to mutation if left unrepaired. For instance, the DNA of a human cell loses about 5,000 purine bases, adenine and guanine, every day because their N-glycosidic linkage to the oxyribose is hydrolyzed. In other words, class, because the N-glycosidic band is destroyed. During hydrolysis, and this reaction and this spontaneous reaction will result to what is known as depurination. And example of purine, in other words, this is the removal of purine in the nucleotide. Just imagine if the purine bases are removed in the nucleotide. It is not anymore a DNA. An example of purine, adenine and guanine. Just imagine if adenine and guanine is removed from the nucleotide. So the nucleotide becomes a nucleoside. It has only now a sugar and a phosphate group. And that is what we call depurination, the removal of purine from the nucleotide. Okay, class, did you understand? Did you understand this depurination? Norisa? Norisa?
Yes po, Doc.
Yeah, did you understand the meaning of depurination?
Yes po.
Okay, what is the meaning of depurination? Norisa? Can you hear me? Norisa, can you hear me?
Yes po, Doc.
Okay, what is the meaning of depurination?
It removal of purine po. In a
Removal of what?
of purine in a nucleotide.
Removal of? What is removed?
Purine, Doc.
Purine. Specifically,
Adenine po and guanine.
and guanine from the nucleotide. Yung removal of purine bases from the nucleotide. That is what is called depurination. This will be a consequence. This will be what will happen if there is no DNA repair mechanism. Later on, you will learn how depurination is repaired. Okay, as you can see, these are the nucleotides. Okay, these are nucleotides, DNA, and the arrow class, you see, this is where alterations of DNA will happen. Or this is where the damage will occur. Like for example, this one. What is this? What is this line? Peter? Peter, what is this line? Peter, are you in? Peter, are you present? Peter is absent. Jem? Taladay?
Sir, Doc, glycosylic bond.
Glycosidic
Glycosylic
band.
bond.
That is the glycosidic band. And you can just imagine if this glycosidic band is destroyed. Okay? Here is also this one. You know what will happen? What is this? What is that? Huh? What is this? Anyone? Anybody, what do you call this one? Anybody?
Sir, peptide bonds po.
This one? Peptide bands? This one? Is that a peptide band? This one? Can you see my pointer? This one?
Amino group.
Amino acid.
That is the amino group. And this is the band that attach the amino group on this carbon atom. What will happen when the amino group is removed? What do you call that process?
Deamination po.
Deamination. Okay, so as you can see the arrow, that is where alteration of the DNA will occur. What is the consequences? Spontaneous deamination of cytosine to uracil in DNA occurs at a rate of about 100 base pair per cell per day. So what will happen to cytosine? When it is deaminated, it will be converted to uracil. Okay. DNA bases are also damaged by encounter with reactive metabolites produced in the cell, including reactive forms of oxygen, or by exposure to chemicals in the environment. Okay, so this is what happened. Okay. The first is what is known as deamination. Okay? Deamination. This is the removal of amino group. Okay? Deamination, spontaneous hydrolysis. The removal of amino group. Okay? This is when this is hydrolyzed. So this amino group now is removed. And so when this is removed, what happened to the structure? This is changed to what? The, 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 when the amino group of the uracil or of the cytosine, when the amino group of the cytosine is removed, cytosine now will be changed to what? It will be changed to
Uracil po.
uracil. You look at the diagram, so this is now the uracil. Okay. And you know, what is this type of nucleotide? What is this type of nucleotide? This is a what? This is what type of nucleotide? This is a what? Michaela, what is the type of nucleotide is that?
Cytidine monophosphate, Doc.
And that is a what?
Pyrimidine po.
The entire. What do you call that? The entire structure. The entire structure is known as what?
DNA nucleotide.
Of course, that is a DNA. And how do you know that that is a DNA?
Because of the
Michaela,
cytosine po.
because of the cytosine. Lahat naman, kahit na RNA, may cytosine. How will you know that that is a DNA? Uh, you've forgotten my explanation on how you are going to recognize, how you will recognize the DNA.
Through its
Angel,
sugar, Doc. The
sugar what?
deoxyribose.
What? Complete your sentence.
Deoxyribose po.
Anong deoxyribose? Deoxyribose? What is that deoxyribose?
In the DNA po, the sugar, the pentose sugar that is in the structure is the deoxyribose
Why is it a
sugar po.
deoxyribose? Why is it a deoxyribose?
Because in its second carbon, it lacks the oxygen at the oxygen po
What is
compared to
black?
the ribose.
Walang blockage yan. There is no blockage. Because in the second carbon atom of the pentose sugar, it does not contain oxygen. That is just the answer. Wala kang nakikita ang oxygen, kaya it is called deoxyribose. Why? Because in the second carbon atom of the pentose sugar, oxygen is lacking. Ganun lang ang sagutan dyan, Michaela. Huwag mo nang pahirapan ang sarili mo. Because of the absence of oxygen in the second carbon atom of the pentose sugar. That's it. Now, when there is now the amination, what happened to that DNA? Angelo, Angel, what happened now to the DNA when there is the amination? Angel, are you still in or you are already out? Angel,
Yes, Doc.
oh, what happened now?
By hydrolysis po, Doc.
Anong mangyayari?
Magre-release ko ng ammonia.
Ano?
Magre-release ko ng ammonia by hydrolysis po.
Now, what happened now to the DNA? Mag-release ng ammonia, walang ammonia dyan. Anong mangyayari ngayon sa DNA?
Di ba yung mutation ng ano?
What will happen to the DNA now? Tingnan nyo mabuti ang diagram. Tingnan nyo ang diagram. You look at the arrow kung anong nangyayari. You should know how to interpret the diagram. Very simple lang. O makikita mo, when there is the amination, now what happened to the DNA? When there is the amination, Angel,
Yes po, Doc.
it is very clear kung anong mangyayari sa DNA. How about you, Alexander? How are you going to interpret the diagram? When there is the amination, what will happen to the DNA?
When there is the amination, the DNA is damaged, Doc.
Oh, it is damaged. Why is it damaged? Para kang para kang nagsumagot ng the DNA is damaged.
When there is the amination, the DNA, the cytosine turns into uracil.
Pwede ba interpret mo ng mahusay? It is not the DNA turns into uracil. It is not the DNA that turns into uracil. What turns into uracil? Pax. Pax, how do you interpret this data? Mayroon pa bang kwan ka na Pax?
Based on the illustration, it shows that the DNA on one side of is and if there is the change into uracil.
Again, Pax?
Since the DNA in the left-hand side of the illustration shows the cytosine, when there is the amination, the cytosine will be changed to uracil.
Will be changed, converted to uracil. So what happened now to the DNA? The DNA became abnormal. Why? How come it is abnormal? Why is it that the DNA is abnormal?
Hello po, Doc.
Yes.
Kasi po, uracil can only be found in RNA.
Because the
So-
uracil could only be found in the RNA.
RNA.
Uracil
Can only be found in RNA. So, parang ma-recognize po ng DNA na may mali. Kasi uracil po yung na-form. May na-form po na uracil.
the DNA itself, by itself, walang recognition ng DNA dyan. The DNA by itself, it is now an abnormal DNA because it contains uracil, which is not supposed to be. Huh? What makes it a DNA? Because the sugar that is being carried, it does not contain oxygen in the second carbon atom, which is a characteristic of DNA. And at the same time also, the DNA also is carrying, instead of uracil, it is carrying what pyrimidine base is being carried by the DNA. is the other base that is being carried by the DNA?
It should be thymine, Doc.
It should be thymine. Okay, that is as a result of the amination. How about depurination? As you can see in here, that the purine-based guanine is removed. It is completely removed. And so you look at what happened to the DNA. The DNA does not contain any more guanine. So it is not anymore a DNA or a nucleotide. You call that nucleoside. And that is not a DNA anymore, because a DNA contains purine bases, nitrogenous base, sugar, and phosphate group. Okay. That is when abnormal condition occur in the DNA as a result of the amination and depurination. Ultraviolet radiation from the sun can produce a covalent linkage between two adjacent pyrimidine bases in DNA to form, for example, thymine dimers. Did you understand this, class? If you expose yourself under the sun, what will happen? The UV light could fuse your two pyrimidine bases. Okay? Could link your two pyrimidine bases like thymine, magpo-fuse yung dalawa, thymine, to form a thymine dimers, or a thymine cytosine to form a thymine cytosine dimers. That is what will happen if you expose yourself under the sun. That will be the harmful effect of UV light. There will be fusion of the two pyrimidine bases to form dimers. And this is what happened. As you can see, the formation of thymine dimer. This is the thymine. Okay. So now, because of that fusion, the DNA, the normal DNA is affected. The normal DNA is affected. If left uncorrected when the DNA is replicated, most of these changes would be expected to lead either to the deletion of one or more base pairs or to a base pair substitution in the daughter DNA chain. Mutation will then be propagated throughout the subsequent cell generations. So, if this DNA abnormality will not be corrected, what will happen to this uncorrected DNA? This will be propagated, or it will be continuously be transmitted from generation to generation, from one offspring to another offspring. Or if we are talking of molecular, it will be transmitted from one cell generation to another cell generation. And this is what happened. This is what happened to the DNA when the DNA undergo the process of the amination. Okay? Again, you have to understand replication of the DNA. And in the replication of the DNA, there are enzymes involved in the synthesis of the new strand. In the synthesis of the new strand. So, in the deamination, what will happen? Cytosine will be converted to uracil. And then what will happen to this DNA? It will replicate. Huh? It will divide. Okay, this is the old strand. Remember the lagging and the leading strand when the DNA is replicated. So there will be, this is the old strand containing the uracil, and this is the old strand containing the normal nucleotides or DNA. What will happen if this will be replicated? If this old strand will be replicated? This old strand, when it will be replicated, the uracil will pair with adenine. Okay? The G changes to A because that is the normal pair of the uracil, adenine. Okay? Whereas here, since this is normal, since this old strand containing the guanine, it will be paired normally, it will synthesize normally, the DNA containing the cytosine. So this is the normal pairing. It is a normal pairing. And usually it involves the DNA polymerase. You have to think of the replication of the DNA, which involves the DNA polymerase. Okay. And then again, if this will be undergo replication again, this DNA, it will divide again, and then the old strand will develop into a strand containing adenine, and adenine, it will form thymine. But this will be the strands containing the uracil will be continuously be synthesized from cell to cell generation. In other words, every time there is replication, the cell will be carrying an abnormal DNA. It will be carrying an abnormal DNA. And then what will happen during depurination? That is when adenine is removed. Okay? Depurination, adenine is removed. So again, there will be, during the process of replication, there will be a split. This is the old strand, and this is the old strand. This old strand, which is carrying the normal DNA sequence, will be replicated by the DNA polymerase to form a new strand which is normal. It is normal because the pairing is complementary. But how about this one? The pairing is normal, but however, the strand will become shorter. The strand will become shorter. And you know the implication of this one? When this will be transcribed into messenger RNA, it will have a short messenger RNA which lack the necessary, which is lacking the necessary codon. And so when the messenger RNA will be translated, it will synthesize an abnormal protein. When this DNA will be translated into protein. When this will be transcribed into messenger RNA, the messenger RNA is abnormal. And that is when the messenger RNA is translated, it will synthesize an abnormal protein. And this setup will be transmitted from cell generation to cell generation. This will be again replicated. And that is how, what will happen if that will not be repaired. The cell all throughout the generation will be carrying an abnormal DNA, an abnormal DNA strand. And so that is the reason why there will be a genetic defect, an abnormal disorder, and a genetic defect, a physical defect, a genetic disorder because of this condition. So now the DNA now could be repaired. The damaged DNA can be removed by more than one pathway. Usually the cells have multiple pathways to repair their DNA using different enzymes that act upon different kinds of lesions. The damage is excised, yung damaged DNA is removed, the original DNA sequence is restored by a DNA polymerase that uses the undamaged strand as its template. And then the remaining break or nick in the double helix is sealed by DNA ligase. And how does it happen? So this is the base excision repair. When you are talking of the base, you are talking of the conversion of cytosine to uracil. That's why it has to be removed. And this could be removed by the enzyme known as DNA glycosylases, which remove the aminated cytosines, removes the aminated adenine, remove types of alkylated or oxidized bases, remove bases with open rings, remove bases in which carbon-carbon double bond has been accidentally converted to a carbon-carbon single bond. Okay, and that is how this DNA glycosylases is utilized. The DNA glycosylases is utilized. Remember that this is an enzyme class. And then the other one is the base excision repair. That is, the entire base is removed. Like for example, in this case, this is the normal double DNA strand. As you can see, class, that this is the normal double-stranded DNA. And then by spontaneous deamination, what happened to cytosine? The cytosine is converted to uracil. So, uracil and guanine, it is an abnormal pairing. So what will happen is that uracil will be removed by the uracil DNA glycosylase. Okay? It will be removed. So now there is a missing base. What will happen? This sugar and the phosphate group, it has to be removed also. And what is that enzyme involved? The AP endonuclease, apurinic endonuclease, remove sugar and the phosphate. So the sugar and the phosphate now is removed by AP endonuclease. So there is now a gap. And how are you going to repair this gap? Now, how will you restore this gap? And this could be done by DNA polymerase. The DNA polymerase will bind on this, and then it will restore the normal pairing of guanine making it cytosine. Okay, so the normal pairing now is restored between guanine and cytosine. So there is a gap here and here, here and here. There's a gap. There's a gap, or there is a nick, and that gap will be repaired by the enzyme known as ligase. And that is how the amination is repaired by three enzymes, by four enzymes: enzyme glycosylase, enzyme endonuclease, enzyme DNA polymerase, and enzyme ligase. An enzyme ligase. Now the nucleotide excision repair. That is how are you going to repair the thymine-thymine dimer, thymine-cytosine dimer, and cytosine-cytosine dimer. Because as what I explained to you, when you expose the DNA under the sun, the UV light will cause a reaction forming the dimer, thymine-thymine dimer, and thymine-cytosine dimers, and how it is being repaired. How will it be repaired? And the process is known as nucleotide excision repair. So this is now how it is being repaired. Okay? This is now the dimer. Okay? This is now the dimer. When this normal DNA was exposed to sunlight, a cytosine thymine dimer was formed. So this is the one. In other words, it joined the cytosine and thymine dimer joined together. There's a link. And so what should be done? The first thing that will happen, that it has to be, the DNA should be excised. And this is by the action of what is known as excision nuclease. Okay, this is an enzyme that cut the DNA. So where is the excision? The excision takes place here in this portion of the DNA, and here in this portion of the DNA. So here what will be left is the cytosine thymine adenine pair, when this fragment will be excised. So by the action of the excision nuclease, this entire fragment will be excised. So this one now, which is carrying the cytosine thymine dimer, tinanggal na siya. It is removed. It is removed. And so, Michaela, what do you think will happen? How this will be repaired? Michaela, how this DNA strand will be repaired?
The DNA polymerase po will pair it up to the correct bases together with the DNA ligase to correct the
the first thing that will happen to the DNA polymerase, it will bind into this DNA fragment or into this DNA strand. It will bind. And then it will synthesize continuously to form a new fragment. Huh? Michaela, there will be the binding of the DNA polymerase to synthesize a new fragment, a new DNA strand from this fragment. As you can see in here now, that there is a newly synthesized DNA fragment. And then the nick here and here, the nick will be restored or it will be repaired by the enzyme known as ligase. And this is how a thymine dimer is excised and it is repaired. And this is how a thymine or acetylcysteine dimer is repaired or it is removed from the DNA strand. And here there are again 4 kinds of enzyme. You know the function of the helicase? The function of the excision nuclease, it will just cut on this. It will cut this fragment, DNA fragment. But again, after cutting, the DNA strand will remain. And how are you going to remove this DNA strand? You can remove that by means of the DNA helicase. Remember the DNA replication? It will open up the DNA. So the DNA will remove. When the DNA helicase acts on the DNA strand by cutting the hydrogen bond. Okay? By cutting the hydrogen bond. Okay. So, do you have any question, class? May mga tanong-tanong ba kayo? The DNA repair mechanism, is it in your assignment? Is it included in your assignment? Christelle, Mariel, Gemella,
Yes, sir.
is this included in your assignment? DNA repair mechanism. Have I given you this assignment?
Not yet po.
No?
No, sir.
Is it? No.
No pa po.
Gem, is it included in the assignment? DNA repair mechanism?
Hindi po, sir.
Hindi yata. It's not.
Yes po, not included.
So, I'm going to post again. I will post the new assignment, which is the DNA repair mechanism. Is Vincent still around? Vincent, are you still around? Sora? Okay. Sora,
Yes. Yes.
do you have access to the assignment? Sora,
No. She was asking me to ask you, what's your email address is?
have you accessed the assignment in the Google Classroom?
No. Yes.
Sora, yes? Vincent,
Yes.
yes?
Yes. Yes.
Okay. Have you posted your assignment? Have you answered your assignment and posted it in the Google Classroom?
Let me ask her. Please wait a second.
Yes? Is it Vincent? Has she posted the assignment? Has she answered the assignment? Has she answered the assignment? Not yet. Sora, do you have your email address? Do you have your email address?
Yeah, she has a Gmail address. And should
That
I
is
send
from
the address to you?
Tua email address or personal email address?
It's a personal email address.
Personal or Tua or university email
Oh.
address?
Tua.
Yes, Tua email address.
Yes.
Okay. Okay. Jem, Jem, do you have the email address of Sora?
Yes, sir. I sent the updated list po sa
Okay.
email niya last week.
You think on the listing, is it there?
Yes,
Okay.
sir.
Okay. Can you send me the Tua email address of Sora? Jem,
Yes, yes, sir.
how about that? Okay. Okay with you, Jem? Can you send me the Tua
Yes.
address
Yes, sir.
of
I
Sora?
will send it po.
Huh? Okay. Do you have any question? Okay, I'm going to see you again next week.
Sir, I have a
And
question.
then I will keep on, I will continue my lecture, and then you will start your report first week of October. Okay? Okay, class, good luck.
Sir.
See you again Saturday.
Sir,
Sir.
Sir?
Sir.
Yes?
Sir, pwede po ba kami makahingi ng copy ng PowerPoint nyo?
Pardon?
Pwede po makahingi kami ng copy ng lectures nyo po?
Ng lectures.
Yes po.
Do you know how to answer the assignment? Alam niyo ba kung paano sumagot ng assignment? I want to see your assignment, huh? Because I would like to know whether you know how to answer that assignment, because that assignment of yours that will strengthen your knowledge about my lecture. Kaya titingnan ko muna yung assignment nyo, how you answer your assignment, kung if it jive with my lecture. If it jive with my lecture, there is no need for you to have my lecture, copy of my lecture. And then either you can take picture of my lecture. Huh? Okay, see you again Saturday.
Professor?
Yes. Benson?
Zora, let me ask you, where should she submit her assignment? To your email address or to the Google Classroom?
You can access that in the Google Class. Okay? In the
Okay.
Google Class.
Okay.
By the way,
Okay.
can Sura really access the Google Class?
Let me ask her.
Can she really access the Google Class? Or have she accessed already?
She has the access to the Google Class.
Okay. There is the assignment there.
Okay.
There's the assignment. Or else, I will ask somebody to send her the assignment.
Okay, I see.
I will ask somebody to send her the assignment. And also about the syllabus. Does Sora has a copy of the syllabus? Benson, please ask her if she has already a copy of the syllabus.
She said no.
No, okay. We will send her the syllabus through
Okay.
her email.
Okay, it's very kind
Yes,
of you.
Abdul?
Sir, good evening. May I ask, sir, if I would like to suggest, I would like to ask Mr. Vincent um, if it's convenient to communicate through chat. I could actually send you an instruction. Perhaps I might be able to record a video on how you would be able to access to attach the assignment.